Hello, good afternoon, everyone. Welcome to the BRAMS CRBLM lecture series. It's my great pleasure to welcome today Dr. Robert Zatorre, and I have a very challenging task to introduce him to say anything new because you can kind of not imagine the neuroscience of music field without Robert. But so in order to find something new, I went to his Google Scholar page, and I actually discovered something quite remarkable. In there you have all these papers that are cited, hundreds, sometimes even thousands of times. And I discovered that Robert has a paper that's cited more times than the year in which it was published. He actually has two of them. And so I'm trying to push for that as a new measure for bibliographic success. So Robert was born and raised in Buenos Aires, and he left the country of tango behind to do his PhD at Brown, and then worked with Brenda Milner, and never left us, thankfully for us. And in 2006, he was a co-founder of this really obscure institute. You might have heard about it, BRAMS, it's called, and it's in the basement here. And basically, he has been like a co-director of this ever since. And he has many prizes, and the latest one, or one of the latest one, I think, is in 2020. He received the De Carvalho Heineken Prize, which is one of the most prestigious prizes in the Netherlands. And I've been curious if that comes with a lifelong free supply of Heineken beer, but I haven't gotten around to ask Robert that. So with no further ado, thank you very much, Robert, for being here with us today, and I'm looking forward to the talk.
They did actually send me, because there was supposed to be a big event in Amsterdam, but of course it was all canceled. So they sent me a box with a bottle of Heineken and some other things. And of course, the bottle exploded and there was not even any beer. Yes, well, thank you very much. Merci beaucoup pour cette belle introduction. Je suis très content d'être de retour de ce côté de la montagne depuis 2 ans, quoi. Et puis, c'est dommage qu'il y a beaucoup de monde qui ne peut pas être ici, notamment nos amis les deux Simonet, qui comptaient être ici, mais ils sont chez eux. Alors, j'espère qu'ils vont bien et qu'ils vont nous suivre par la voie virtuelle. So let me just mention that. So the talk I decided I would give today is focused on a particular topic. Can we actually have the first slide, please? And it's a topic that I've been working on for ever since I started, really, which is the specialization of the 2 hemispheres for processing of auditory stimuli. And this particular talk was given in Paris last fall at the Fondation pour l'Audition. And so it's sort of oriented around that topic. And when I give this talk, I use the sort of motto, if you will, of the Fondation pour l'Audition, which is kind of cool. It's à l'écoute de la vie. And I think it's a very nice phrase to capture the importance of sound in our daily life. Now, if we think of that idea in a more kind of broader biological context, c'est quoi la vie? That depends on your species. So if you are a bat, for example, your auditory world is very specialized, and you make extremely high frequency sounds that you use to navigate and to find insects to eat and things like that. And if you are a member of the avian, one of the many avian species, you have a totally different set of sounds that you produce and that you perceive, and that are very important for communication and for finding a mate and for defending your territory and things like that. So in that context, what is the communicative system that we humans have? Now, the majority of people, probably not those of you in this room are already indoctrinated, but the majority of people, if you ask this question, they would say, well, it's speech, right? That is our communicative system, because we all talk and we use it to communicate and we use it to defend our territory or sometimes to attack other people's territory, if you're Mr. Putin, and so on. But of course, I think many of us would argue that this is not the only communicative system that we have. We also have music. And that's, I would argue, that's maybe one of the main points of this talk, is the argument that music constitutes a parallel and distinct communicative system, distinct from speech. It has some very important and interesting commonalities, but I think from the earliest level of processing, it uses a separate channel, and that's what I'm going to try to convince you of. And I'm going to convince you of that, or I'll try, by focusing on the functional properties of the human auditory system and the differences between the left and right auditory cortices. So very roughly speaking, the auditory part of the brain is found on the superior temporal gyrus here. To that pointer that we have.
Is it here, this one?
Yes, thank you. So in the temporal lobe here, I don't know if you can see the pointer. In the temporal lobe here, we have these colored areas, and they represent different subfields of auditory cortex in a kind of a cartoon. And we know, of course, that this system communicates broadly with the rest of the brain via a number of different connections, but we'll mostly be focusing on the auditory cortex itself in this lecture. And the reason for that is because it's been known really since the time of Wernicke, for example, in the 19th century, that there are different functional properties between left and right. And in our own work, very early, back in earliest days of neuroimaging in the 1990s, we saw some evidence of this sort of distinction as well. So if we looked at brain activity of speech compared to acoustically matched noise, we would tend to see greater response on the left. And if we did the same manipulation, we would tend to see greater response on the right. Although in both cases, there's always a bilateral response. But this is a very simplified view, and we didn't necessarily have a good model to explain why this might be the case. And that's, I think, the job that we've had over the last 30 years now, is to try to understand why these sorts of asymmetries might emerge. And there are different ideas behind it, but a lot of the work has been devoted to understanding the hemispheric specialization for speech, because more people in general work on speech in the scientific world. But of course, we focused a lot more on the right side and what it may be specialized for. One of the first hints we had in terms of specialization was from this early lesion study that was carried out by Ingrid Josrud, who's now a professor at Western, and with Virginia Pennion, who's, as you know, a member of BRAMS and is at Concordia. And actually, Virginia developed a way to measure the size of the lesion. And we were able to identify patients whose lesions were including a portion of the auditory cortex that's very close to the primary region. So probably, we go back to this diagram, probably in this sort of green area here. So probably part of belt cortex, physiological terminology. And what we did was then a very simple psychophysical task. You hear two tones, and you have to say if they go up or down, and then we titrate the size of the difference between those tones, a standard staircase procedure, and we then measure the threshold. And the interesting finding is that in this group of patients who had this kind of lesion on the right side that encroached into this particular auditory region, their error rate for this task was much higher. That is to say, their threshold, they needed a much higher threshold to respond than control participants. This was also true for patients with other kinds of lesions, especially the ones on the left. And in fact, the ones with the equivalent lesion on the left were marginally better than controls, which is sort of remarkable. So this is one of the first hints that we had that maybe part of what's going on here is that this region, the sub-region of right auditory cortex, is important for the representation of very fine-grained frequency information that would lead to the ability to perceive fine pitch differences, which from the beginning, we always thought this is really pretty critical for music, right? So this is sort of the initial conclusion, but we wanted to go a little bit further than that, and maybe to look at, is there some kind of difference in resolution in the frequency domain, and does that map onto the two different hemispheres? And there are several different studies that we've done over the years that speak to this. One of them was done by our dear late friend Krista, who sadly is no longer with us, and we miss her so much. But what Krista did in this study is manipulate the size of the pitch intervals in a systematic fashion of this little up and down pattern. And what she found was that the greater the increase, the more the right auditory cortex followed the increase. So there's a sensitivity here to the pitch variation. But a very interesting part of the study, which was published in 2008, along with Isabelle Peretz, of course, was that it's not that the left side didn't respond at all. It's just that the left side didn't have this kind of quasi-linear relationship. If you look at the left side, what it does is it basically doesn't respond at all until the difference is large. So it's just not sensitive to small differences. It does respond, but it takes a big difference for it to be able to give a response. So this, we think, is really kind of a critical finding, because it supports this idea that one of the relevant differences between left and right is in terms of the resolution that the two systems have. Now, we can test that in various ways, and a very recent test of that is provided by my former PhD student, Reiko Matsushita. And what Reiko was interested in was looking at these very, very fine-grained differences. And to do that, she adapted a paradigm that we had done a few years earlier, where we create what we call micromelodies. These are melodies that are in between the cracks of the piano. So they have very, very small frequency differences. And we can vary these differences in terms of the logarithmic unit, which is cents. So most of you probably know that a semitone consists of 100 cents, which are logarithmically equally spaced differences. So 60 cents is really pretty easy to perceive. I'll tell you a little micromelody. You can definitely hear that's varying in pitch. Here's one of 30 cents, which is much smaller. But you probably can still hear that. If you can't, you should talk to Isabelle and go to the Amusia lab. And here's one with 10 cents, which is pretty much close to the threshold. There's a missing note there, sorry, but you get the idea. And the task was using a brain stimulation technique, transcranial direct current stimulation, because the idea was to see if we could test the effect of the stimulation on the right versus the left. And it's a rather complicated experiment because we did both brain stimulation and recording of brain activity using magnetoencephalography. And our colleague Sylvain Baillet was a little alarmed when we told him we were going to bring in 2 wires into the MEG scanner, which is hypersensitive. But it can be done safely, and it does produce artifacts, but they can be removed in post-processing. So the task basically was a learning task, and I won't go through all the details, but it extended over 3 days. And the manipulation was that each of 3 different groups received either stimulation to the right on day 1, or stimulation to the left, or a sham condition. And they of course, didn't know which was which. And then we tested their performance on days 2 and 3. They get feedback on every trial, and it's a discrimination task to, you hear the first melody and the second melody, you just have to say if they're the same or different. And normally people are able to learn this. So to measure it, we simply calculated the psychometric curve and determined the threshold for a block of trials. This is standard sort of psychophysics. And then we use that threshold measure as the dependent variable to look at learning. And so if you look here at the sham group, which is shown with the dotted lines, we've normalized everyone to 0 for day 1. And as the days progress, they get better and better. That is, the threshold drops, right? Now look what happens when you stimulate the left auditory cortex. Absolutely nothing. It has no effect. But when you stimulate the right side, the subjects actually don't learn, and they actually get marginally worse. So it's a very significant difference here. Remember, the TDCS is applied on day 1 in the middle of the 3 sessions that day. So this is good causal evidence that when you disrupt the functioning of the right auditory cortex, it interferes with the ability to perceive these very fine-grained differences. Now, what's even nicer is that because we were able to measure the MEG responses, we could look at the size of the evoked response. So here we're just measuring the magnetic N1. It's a standard first evoked component. And what you see is that if you stimulate the left, the N1 goes down and then recovers. If you stimulate the right N1 goes down and then recovers. So it's not that when you stimulate the left auditory cortex, it doesn't affect the brain, right? It definitely inhibits the response onto the left side. It's just that because the left auditory cortex doesn't really do very much for you with pitch, it doesn't really matter that it's been affected, right? It has no particular effect. Whereas on the right side, it has a sort of equal magnitude change, and yet this effect is responsible for the disruption, and we show that by looking at the correlation for each individual. Each dot here represents an individual. So the degree to which the N1 is affected on an individual basis by the stimulation is predictive of the degree of learning. So the more the N1 is affected, the worse your learning is. And that's not true on the left. Like I said, it's not that the left auditory cortex is not affected by the stimulation. It is, but it just doesn't contribute to the task. So we think this is pretty good causal evidence in favor of this sort of fine-grained distinction. Further evidence comes from looking at the frequency following response. This is work done by my former PhD student, Emily Coffey, who's now also at Concordia. And most of you are familiar with the FFR, so-called, with EEG. That's the usual way to do it. But with MEG, you can also get it, and it's this potential that, as its name suggests, follows the frequency, follows the fundamental frequency. And the cool thing about doing it with MEG is that you can then see where the source is located in the brain. And when we plotted this, we found, somewhat surprisingly, but very rewardingly, that there was a much bigger response on the right than on the left in terms of magnitude. And here's the distribution across all the different individuals. You see that almost every individual had a right greater than left response. Only 2 of them actually went the other way, and no one had a large discrepancy. And importantly for our story, only the magnitude of the potential on the right, but not on the left, correlates with frequency discrimination behavioral performance. So people who have good frequency discrimination are the ones who tend to have a large frequency following response from the right auditory cortex. That relationship does not hold on the left. So we can now add to our conclusions that there is this difference in terms of fine-grained processing and resolution. But now we still need a mechanism. We still need to understand what actually explains this difference in resolution. So we've been able to define the effect better, but we still don't know the cause of it. And to do that, we go back again in time a little bit to around the turn of the millennium, when Pascal Galin, who's now a professor in Marseille, and I did these experiments where we tried to look at not only the frequency side of the equation, but also the temporal side of the equation. And these are the two dimensions of sound, right? You can vary the spectral features, the frequency content, or you can vary the temporal features. And so in this experiment, we devised this stimulus set where, on the one hand, you start with a sort of slow alternation of tones, and then they get faster and faster and faster. So this is the average duration of each element in this set. And when we do that, and we look in the brain, we see these two green blobs, and the green blob on the left is larger than the one on the right. But if we take the same two tones and now keep their speed constant and change the size of the frequency, average frequency difference from, we started here with an octave, now we have a half octave, a quarter, and so on. These are very, very small differences, right? When we look at that, we see these two red blobs. Again, they're bilateral, but the red blob on the right is much higher than the one on the left, or not much higher, but significantly higher. So here we have a nice double dissociation, right? The response on the left is more sensitive to temporal differences than to the spectral differences, and the one on the right has the opposite. And in fact, this sort of finding has been, after we published our paper in 2001, several other groups did similar things. Mark Schrimwiesner's group in Leipzig, and this is from David Popo's group, and this is from Tim Griffith's group. Several different types of manipulations. I won't go into the details, but they all find sort of similar patterns, or patterns that I would say are compatible with this idea that there is greater sensitivity to spectral types of features on the right and temporal types of features on the left. And we had already, with Pascal and Virginia, we had already kind of proposed this theoretical idea. And this is what we said back then, that we argued that the auditory cortices in the two hemispheres are relatively specialized, such that temporal resolution is better in left and spectral resolution is better in right. So that was sort of our theoretical position in 2002. And so we have, I think, substantial evidence in favor of that kind of model that we had proposed, but we're still lacking the mechanism. So, the first step was to identify where those differences might come from. The second step was to try to understand what might underlie them. But that doesn't mean you have a physiological understanding, right? It's still kind of descriptive. So what I'm going to present to you in the rest of the talk is two pieces of evidence that tell us something about what might be the underlying mechanism. The first piece of evidence has to do with connectivity profiles. And this is work done by Kuok Cha, who was a graduate student with in my lab and with Mark Schrimwiesner when he was still here at BRAMS, actually, back in the good old days. So what Kuok did was, first you take this sort of standard frequency map. This is a map that you're all familiar with. So the color scale simply represents the frequency that each voxel is most highly tuned to. So if you see yellow or red, it means their voxels tuned to relatively high frequencies. And if you see blue, it means that their voxels tuned to relatively low frequencies. And this is standard, right? This is what everyone always knows. But we can do something else, which is to look at the functional connectivity map. So this map is telling us something different. It's telling us how, for any given voxel, how much is it connected functionally basically how well is it correlated, how well is its activity correlated over time with other voxels. So you see these little crosshairs here. So if we take that little target voxel there, what is shown in the yellow are other voxels that are highly linked to it in both hemispheres. And if we take this other target voxel here, or this third one over here, you see that the patterns are quite different. Okay. Now we have two independent bits of evidence, one about the frequency content and the other about the degree to which each voxel is correlated. We can put them together in the following way by ordering the connectivity measures. So here we have connectivity measures on the y-axis, and now we are ordering them as a function of the frequency of the different tones, because we have both pieces of evidence, right? And to make a long story short, you can ask me questions if you are interested in the technical details. What we end up with is a plot like this that tells us what does the connectivity look like as a function of the frequency difference of the tuning of the two voxels. And you see that it isn't flat, it's sloped. What that means is that voxels that are tuned to the same frequency, where the difference is 0, have the highest connectivity. And voxels that are tuned to large frequency difference have the lowest connectivity. In other words, the voxels that are tuned to the same frequency are talking to each other more than the voxels that are tuned to different frequencies. And we see that, this is the punchline here, is that we see that effect much more strongly on the right than on the left. So on the left, it's much flatter. It's still there, but it's much flatter. On the right, it's much steeper. So what that means is that this phenomenon of communication as a function of frequency is a mechanism which is much more important on the right, whereas on the left it's flatter, which means that voxels tend to talk to each other independently, relatively more independently of their frequency tuning. And what that means is that they integrate across frequencies because they're talking to each other even though their frequencies are different. Whereas here, they're segregating across frequencies because they're talking to each other only when they're similar. And this is actually relevant to some physiological studies that I won't go into the details of, but this physiological study done in rodents in 2007 found that they said that the excitatory intracortical input was very highly tuned to frequency, and this is attributable to a selective amplification of cortical response at preferred frequencies. So this is the physiological counterpart. It's basically saying that if you have voxels that are located wherever and they're all tuned to the same frequency, by integrating their response, they can sharpen their sensitivity. So that's the first bit of physiological mechanism that I want to propose. And the second one has to do with spectrotemporal modulation. And so this is a concept that has grown a lot in importance in auditory neuroscience over the past 20 years or so. And it was sort of promoted by a number of people. Frédéric Duquesne, who is in San Francisco, who is an ethologist and works a lot on birdsong, and Shihab Shamma, who is somehow in Paris and in Washington, possibly at the same time. Maybe there were two of them, and that's how because he publishes, 50 papers a year. But he's a neurophysiologist, and he's done a lot of very important work here. So we're all used to this spectrogram, right? Everyone knows what a spectrogram is. It's the energy contained in a sound plotted in terms of frequency as a function of time. This is just some kind of speech pattern. What's interesting is that you can perform mathematical operation on this spectrogram to convert it into what sometimes is called a modulogram, or sometimes people call it a spectrotemporal modulation plot. It goes by different names. But basically, what you're doing is you're decomposing this sound. You take the entirety of the sound, and you one way to think of it is that you filter it according to different spectrotemporal filters. So for example, if you have a piece of sound, there's a little oval there that you can't see very well. If you have a piece of sound that has a lot of harmonics, but is relatively steady state, it would look like this, is sort of horizontal. And so that gets plotted over here on this graph because it has relatively high spectral modulation, because there's a lot of harmonics per octave, but it has nearly zero temporal modulation because it's steady state, it's not changing at all, right? Whereas over here, you have a sharp onset over here, right? This sharp onset, it represents a relatively high temporal change, but it represents a relatively low spectral change because, as you can see, it doesn't have much harmonic structure or spectral structure. It doesn't have to be harmonic. And then you have intermediate cases like these little stripy things, which are somewhere in between, where there's both spectral and temporal modulation. Now, why do we care about doing this complicated mathematical transformation? Well, the reason is because, as Shama showed, when you look neurophysiologically in the brain of many different species, including birds, including primates, and I'll show you, including humans, what we see is a joint sensitivity to both time and frequency. So this is basically a receptive field, a spectrotemporal receptive field. And what it's telling us is that this particular neuron likes to respond to events that have a given frequency and that have a given time frame. So this particular one has a particular joint sensitivity to frequency and to short temporal events. And what's cool is that this can be transformed via Fourier transform into this modulogram. So this thing here, this spectrotemporal response function, is formally identical to this. So it's temporal modulation on the x-axis and spectral modulation on the y-axis. So in other words, when we go from the spectrogram to this weird representation that we're not so used to seeing, we're actually much closer to the physiology of the brain. If any of you have ever studied visual neuroscience, you will be familiar with this concept because it's essentially identical to visual gratings. You may have seen work where visual scenes decomposed into different spatial frequency components that have different orientations, right? And so this is essentially the same thing, except in vision, the dimensions are, vertical space and horizontal space, whereas in hearing, the two dimensions are time and spectral axis. So I mentioned that this was recorded. I think this comes from a cat study. I'm not sure, maybe a ferret. Shama does a lot of work with ferrets. But with Mark, we wanted to determine if we could show evidence for spectrotemporal receptive fields in humans. And it's not because it's not so interesting to show that humans are like monkeys. They almost certainly would be. But what's really important is to see whether we can even pick it up with fMRI. Because those physiological studies, they put one electrode into one cell. But in fMRI, we're looking at thousands and thousands of neurons all at the same time. So to do this, Mark devised this set of stimuli, which basically are like these things here. So each stimulus has a particular value in both temporal modulation and spectral modulation. So for example, the one at the bottom corner here, and I'm going to play these for you, they will remind you a lot of like Space Invaders. If any of you played that game, or you're all too young for that, but it's what I played when I was younger. So that sound is relatively slow moving and relatively harmonically not so rich. Here's one that has the same rate of change in terms of speed, but is harmonically richer. Okay, and then here's one that is very fast. That's when the aliens are attacking you. And then this one has both high spectral and temporal change. Okay, so we probe. This is a horrible experiment to be in, because you have to listen to these things, right? This whole matrix of 64 stimuli for, I don't know, like an hour. But so each brain voxel is probed with each of these 64 stimuli, and then we plot the response profile for each voxel. And what we see is that each voxel does have a particular preference. In this case, for example, this particular voxel really likes sounds that have that particular combination of spectrotemporal modulation. That's the highest response. It doesn't like very much the ones that are down here, and sort of has intermediate values for the ones down here. So here we see sensitivity to spectrotemporal modulation, a lot like what you see in those animal studies, because it looks not so dissimilar to this. But we're looking at a much more coarse grain of spatial resolution, because we're using MRI, right? We are not using electrodes. There are some studies that have come out subsequently that do use implanted electrodes in humans, like with epilepsy patients, and have been able to document these sorts of responses, with much greater precision than we were able to. Here are, just a plot of the different kinds of responses that we get. So there are some voxels that really favor like one particular combination of spectrotemporal. There are others that really are tuned to particular spectral ranges. Like here's one that likes high spectral resolution, or low spectral resolution, or intermediate, but doesn't care very much about the temporal. And then here's some others that care a lot about the temporal, but don't really care very much about the spectral. And then there are other classes of neurons that are more complex, that have like even two favorite spots. So there's, it's kind of a zoo of different responses. But the important thing is that we can actually dissociate responses that are more temporally tuned and more spectrally tuned. Okay, now keep that in mind, because the reason we care about these spectrotemporal response functions is, on the one hand, because they're physiologically meaningful, but on the other hand, because it takes us back to the very beginning of the talk, which is our interest, I think our shared interest in understanding how music might differ from speech. So how does music differ from speech? Well, again, we've all seen these kinds of spectrograms. This is just sort of randomly selected from internet sources, Mandarin and French speech. So what happens when you represent these things in terms of these spectrotemporal plots? Well, they look like this. Okay, a lot of stuff going on down here. In the Mandarin, you have a little bit of extra stuff up here. Now contrast this with music. Here are two of my favorite composers, Astor. This is Oblivion. You may be familiar with that piece. And this is the Bach Passacaglia Fugue in C minor. You see the pedal part first, and then you get all the rich harmonics. When we perform the same operation on the music, it looks really different than the speech does, right? And it looks really different in a very particular way. Namely, the speech is really exploiting the temporal modulation. There's not much going on with the spectral modulation, except a little bit here in Mandarin, which of course is a tonal language. But compared to the music, there's not much going on here with the temporal. It's a little bit more going on here for the piazzolla, which has some really fast bits, but most of the action is spread out along this axis. Other people have noticed similar things. In particular, Nai Ding and colleagues from David Popel's lab had already noted that speech tends to be faster. So if you look at, this is temporal modulation, speech tends to be modulated at the rate of around 4 to 6 hertz, which is of course a syllable rate. As we're talking, we go blah, 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 blah. So the amplitude modulation there is around that rate. But music, by and large, tends to be slower, which is, I think, consistent with this this is not an experiment. This is just, some pretty pictures, right? But you see here, the music tends to be very slow modulations. Now, I can hear somebody who's saying, yeah, but Piazzolla, Bach, that's all Western music. And what you're talking about is just an artifact of Western music and so on and so forth. So to test for that, we have some very exciting, completely new data that this is literally from like 2 days ago, where my former postdoc, Philippe Albouy, who's now professor at Université Laval, and Sam Mayer, who is at Harvard, although he manages to live in Westmount. We got together, and they're both proud papas. It's sort of cute that they always pose with their babies, who are, I've met them, they're super cute babies. And so we got together because Sam has this wonderful database available of recordings from 21 different societies all throughout the globe, including extremely exotic places like Toronto, and other places like Papua New Guinea and remote spots in Africa or in Finland, or Quechua people in Bolivia. And what they did was they asked people in each of those different societies first to sing a song and then to say something. So super easy, right? And everyone in all these societies, they immediately understood, all societies have songs, they know what that is. And so they sang them and they all spoke. This is part of an experiment where he was also interested in looking at infant directed speech, but we're not really considering that here. So we have these recordings of music on the one hand and speech on the other, or vice versa, music and speech. And so what Philippe did was he put it through this spectrotemporal modulation framework and then did some statistics to compare the music versus speech. And he did that for each of these different societies. And it looks like this. This is just a t-test of the difference between music versus speech in the spectrotemporal plots. And you see these red blobs, which are corresponding to areas of higher spectral modulation for the music. And we put them all together across all the different societies. We see this pattern, which is pretty remarkable because there's this huge overlap right in this spot. Now, there's some of these separate cultures. There's some interesting differences here in the temporal domain, but they all basically agree that when you sing a song, you're using this portion of the spectrotemporal space across completely unrelated cultures that have nothing to do with each other. So I'm extremely excited by this because I think it's nice proof that there is something about music which is not unique to any particular culture, but that has to do with the exploitation of information in that spectral domain. Okay, last piece of the puzzle, which is maybe in a way it's the best piece, is to link this back to the brain, the spectrotemporal business. So to do that, we used, again, going back to this fellow Tunisian, we used an approach that he developed where once you have taken the sound and put it into spectrotemporal space, you can filter it to remove the spectral information selectively while leaving the temporal information alone, or vice versa, separately take out the temporal information and leave the spectral information. And this is what we did with Philippe and Benjamin Morbihan, who's in Marseille. And so essentially you take the sound, and I'll tell you what the sound is in a second, you convert it into this space, which is what you've seen now for a while, and then you remove most of the temporal modulation and leave the spectral information. And then if you convert it back to a spectrogram, it looks like this. So it's basically taking the original spectrogram and kind of smoothing it. It's like you took a brush and smoothed it in the in this axis, right? So you're blurring the time, but you're keeping the frequency. Or you do the opposite manipulation, you leave the temporal information, but you remove the spectral modulation. And this is, now you're smoothing in this direction. You're rubbing out, you're removing the frequency distinctions while keeping the temporal distinctions. So we did that with a bunch of songs that we created. These are really wonderful songs, as you'll see in a second. They've made the top 20 list many times. The way we did it was basically by getting a composer to write us 10 different little melodies that have the same rhythm. And then we matched each of the 10 melodies to 10 different sentences. So we end up with this matrix of 100 songs, 10 sentences sung, each of them to 10 different melodies. And that's cool because it means that we can totally separate the speech content from the melodic content, right? Because we have all possible combinations. And then we apply these degradations. So for example, here's a little one of our favorite tunes. It sounds like this.
Je voudrais parler à mon bon copain.
We hired a very nice soprano who spent many, many hours singing these dreadful songs. And then what happens when we degrade the spectral information? It sounds like this.
Je voudrais parler à mon bon copain.
So it's kind of like Darth Vader.
Je voudrais parler à mon bon copain.
Here is the maximal spectral degradation.
Je voudrais parler à mon bon copain.
So you notice the melody's gone, right? But you can understand the speech perfectly. This, by the way, is also the principle behind cochlear implants, where only temporal information is transmitted, not only, but temporal information is preserved very well, but spectral information is not good. And that's why people with cochlear implants can understand speech pretty well. Here's the opposite manipulation with a different melody here, for example. This is the original.
The people thought a coin was hidden there.
Here's what happens when we remove the temporal information. Really can't get the speech anymore, especially in the most extreme version. But the melodic line is still somewhat preserved. And so when we do that behaviorally, this is what we get with the melodies in speech. Melodies and sentences are shown in orange versus blue. When we do the spectral degradation, you see it has absolutely no effect on sentence comprehension. It just doesn't change. Whereas it really goes down as you remove more and more of the spectral information for the melodies. And the exact opposite pattern for the temporal degradations. The melodies are not affected at all. In fact, they even improve a little tiny bit. Whereas the sentences are very hard to understand as soon as you remove the temporal information. So this is actually, these data are for francophone speakers, and these data are for English speakers, and we see the exact same pattern. And again, people look at this and go, yes, yes, but all of you, Eurocentric people, all you look at are European languages, blah, blah. So we turn to my former student, Yi Du, who's now a professor at the Chinese Academy of Sciences. And she was able to create Chinese versions of these crazy songs. And we see the identical pattern, just a few minor differences, but basically with Mandarin speech, you have the identical pattern of behavioral responses. So we're pretty confident that it's not particular to any one language, although we haven't tested it in other languages beyond Mandarin, but of course Mandarin is a tonal language. So now you're wondering what's going on in the brain. Well, here is, we did that experiment, the behavior of which I just showed you in the fMRI scanner, and we then apply a multivariate pattern classifier to try to classify sentences versus melodies. And we see that the sentences are best decoded from left auditory cortex, whereas the melodies are best decoded from the right auditory cortex. And when Philippe sent me this picture, I had a visual image of this one, right? It's like, where have I seen that before? The difference, of course, is that then this is just sort of phenomenology. We were like, oh, look, there's something on the left and something on the right. We didn't really know where it came from. I think now we see very similar effects, but we have a much better idea of where they arise. And the kicker is that when you look at the effect of degradation on the brain activity, we see exactly what we would expect. So this graph is a little hard to follow, but if you look in the left auditory cortex, which is able to decode the melodies, the sentences, right? If you degrade the information temporally, the classifier no longer can do so. Okay, but when you degrade spectrally, it's perfectly fine. For the melodies, the left side isn't very good at it in any case, so it's not really affected much because it's really pretty poor at decoding melodies. At the right auditory cortex, we have the exact opposite pattern. When the melodies are degraded temporally, decoding is still pretty accurate, but when you degrade spectrally, it is no longer possible to decode them. And of course, the right side is not very good at the melodies in the first place, so not much effect there. And the final little bit of evidence, I won't go through it in the interest of time, is relating the confusion matrix from the classifier to the confusion matrix from the human participant. Because in the previous slide, I only showed you the percent correct, but it's also interesting to look at what are the errors like? Are the errors similar between the way the brain is doing it and the way the actual whole human, whose brain presumably is giving the answers? And the answer is yes. I won't go through the details, but basically there's a significant relationship between the confusion matrices here. So what are our conclusions? Well, I've shown you evidence that these hemispheric asymmetries are dependent upon differential types of processing, in particular that the left and right sides favor spectral integration, temporal resolution on the left, and spectral segregation and temporal averaging on the right. I also showed you evidence that the local connectivity profiles might provide a physiological mechanism for this specialization. Then I showed you that music versus speech tend to exploit different spectrotemporal ranges, as we saw from just this sort of illustration. And then finally, I showed you that these different spectrotemporal ranges actually map onto these left and right differences. So we think that this is, that we've actually made some progress compared to 30 years ago. And that what we're seeing is that there are two parallel systems that are optimized for these two main auditory communication systems that I talked about at the very beginning. Other creatures tend to have a single communication system. We have two. How do we keep them straight? Well, I argue that the way we keep them straight is by using different acoustical aspects of sound to keep them separate. And we then are able to efficiently process them via these two separable mechanisms on each side. And this provides us with a very both sort of ecologically efficient way of dealing with it, and helps us solve the problem that if you have neurons that are specialized in one domain, they can't simultaneously be specialized in the other. Because if you're going to have high temporal resolution, you need to integrate across spectral. And if you're going to have high spectral segregation, you need to average across temporal. So you can't have it both ways. There's the trade-off between them. But you can have it both ways if you have two quasi-independent systems, one housed in each hemisphere. And so with that, I'll make sure to thank all the many different people. I think I've mentioned most of their names along the way. And of course, all of the funding agencies that have made it possible. And thank you so much for your attention and for your kind invitation. It's really a lot of fun to be back en présentiel.
Thank you, Robert, for his wonderful talk. I'd love to invite some questions from both the audience here, but also from those who are following us live through Zoom. I think we have about 30 folks. If you want to ask a question online, please use either your camera and you can ask your question. I think we can put you on the screen here as it might already be happening. You can also put it in the chat, in which case it will be read aloud with all the risk that entails. With that, I think actually Isabel would be the first person. Please, Isabel. Oh, Isabel, I think you're muted.
Sorry, Robert, to be remotely connected. I don't know where you are saying hello. It's so awkward. So I cannot resist. I mean, lovely talk first. I mean, I haven't heard it, so it's all some part I really knew. The physiological code is really impressive. So I really enjoyed this part. I enjoyed the whole talk, of course. And now I will play the devil because, you know, I completely agree with your perspective. I even go further in terms of specialization. And you know, the big issue, and now it's still there, it has been on for a long time, it's the overlap. So how do you respond to the idea that, you know, language and music have so much in common, that they share processing and overlapping brain areas? How do you respond to that question?
Yes, thank you. Thank you, Isabel, both for the kind words and the incisive question. I think it's a very, very fair question. I think there are sort of two answers. One is that although I've emphasized obviously the differences between on the acoustical level between the two, it's nevertheless the case that in many instances both kinds of cues are present. So some of the overlap may be due to that, and also the fact that the hemispheric difference, as I've sort of mentioned on several occasions, is relative and not absolute. So we very often see some response on the other side, just not as clear, not as strong, not as accurate. But the second answer, which I think is probably more important, is that in this talk, I've really focused only on the input level, if you will, right? So I'm essentially talking about the bottom-up side of the equation, the way that incoming information is analyzed and encoded. But there's another huge part of the story which has to do with the top-down component. So as I think I mentioned at the very beginning, the auditory cortices are, you know, the final stage for auditory inputs, but it's the beginning stage of cognitive processing, and information that arrives in auditory cortex is then sent back and forth through multiple dorsal and ventral streams to all kinds of other brain areas. And that, I think, is where a lot of the overlap arises, because once you get beyond the this relatively early analysis level, where you're closer to the stimulus features, to the acoustical feature space, once you get beyond that and you are dealing with more abstract representations that are not so closely tied to the lower level, then you might start to make demands on on systems that are not so specialized, that are more global. So, for example, already at the level of, for instance, working memory, you might have some mechanisms that are in common. At the level of higher order organization, like syntactic organization, you very likely to have some aspects of that complex processing which is in common. And so that's probably how I would answer that question. Although even then, I do think that there's still some segregation, because even like those examples I just gave, and as we know from your own work, auditory working memory, for instance, in people with amusia who tend to have disconnection of the right ventral pathway, as you and Krista showed many years ago, they tend to have much worse working memory for tonal information than for speech. And similarly for like syntactic processes, even though they're somewhat shared, we also see some evidence of hemispheric differences, like as you well know from, like Stefan Kosch's work with the early anterior negativity that tends to be a little stronger on the right for musical syntactic violations compared to language syntactic violations. So I think my guess is that as you proceed from the early stages to later and later stages, the hemispheric differences go from being very strong to being smaller, and possibly at some ultimate stage being completely absent. I don't know if that convinces you.
I think we can discuss further at some point, but I don't want to hold you for too long. But I think attention is very important. It is a top-down aspect that is dynamically developing over time. But this will be for another discussion. So, yeah, thank you.
Thank you, Isabel, for your question. Are there questions also in the audience? We have more questions on Zoom, but I want to make sure we don't forget about the people who are actually here. Yes.
Do you see any like trends with handedness with respect to like lateralization as we do?
Yeah, the question is whether we see any trends with handedness, and I wish I had an answer for you, but I'm afraid we don't. We have not studied it. Hardly anybody has studied it. It's sort of a shame. But it's, if anyone's looking for a PhD thesis topic, go wild, because there's hardly anything that's been done on that. And I think it would be quite interesting to see, because one of the claims that I'm making is that this hemispheric division of labor is more efficient. If that's true, then individuals whose hemispheric difference is less sharp should show some cost of that. That's a prediction that my model would make. And I don't know of any evidence that would support that. So maybe if you look at people with mixed handedness, for instance, that would be interesting to see, but there's not much out there.
Thank you. Let's move to Christina der Nederlanden on Zoom.
Hello, Robert and everybody. Really nice talk. Thanks so much for the good, like Reader's Digest version of all your work. I really appreciated it. The new data that you were presenting that you said just came out a couple days ago was something I was kind of anticipating to see something more like your last summary slide, where you kind of saw for speech a really horizontal, a lot more temporal information, and for music a lot more spectral variation, and you were showing something about overlap. So I think it's actually more of a clarification question. What am I looking at on those figures, and what is it telling me about overlap or distinctness in terms of spectrotemporal modulation?
Yeah. So thank you. The point I was making was that there's one part of the spectrotemporal space that overlaps across all these different cultures. And that is an area of very slow temporal modulation and sort of moderate spectral modulation. That's the one place where things overlap. There are other places where individual musics seem to be different. So I'm not saying that they're all identical. I'm saying that they have one thing in common, and that one thing that they have in common is that they all seem to agree that that area of relatively moderate, I would say, spectral modulation and relatively low, that is slow, temporal modulation is characteristic of music as determined by the very people in those places. One thing I like about this study is that, the experimenter did not go to those places with the stimuli and say, hey, listen to this and what do you think, right? This was the people from those cultures who were asked to sing the song. So it's completely spontaneous on their part. It's what they think is music. It's not what the Western experimenter tried to impose. I think that's very important. So I'm not sure if that answers your question. There's one other detail of that experiment, which is that people were asked to either speak or to sing. So we do not have any recordings of music generated by anything other than the voice. And all the songs, of course, have speech content in them, right? So we can only look at what differentiates song from speech. We cannot look at what differentiates speech from music, because when we look at speech versus song, there's nothing left because both of them contain speech. So the temporal modulation side is going to cancel out, right? Because in both cases there's speech. So the only thing that we can look at in this particular sample is what differentiates, what makes spoken vocalizations musical. And what makes them musical is that they tend to have low temporal rate of change and moderately high spectral modulation.
Thanks. That was a perfect explanation. I appreciate it.
And when I said it came out like 2 days ago, what I mean is it came out of Philippe's computer 2 days ago. So don't look for the final paper yet. Philippe has another paper that just came out today, which is also his birthday. So it's kind of a nice birthday present, but it's got nothing to do with this. So we're still chewing on it, and we have a lot of other analyses to do, but I'm really excited about it, and I wanted to share it with you.
Do we have more questions here locally in the room? Yes.
Thank you. It was really a great talk. I'm wondering if there are what you would think, or if you've looked at differences with musicians and non-musicians, and specifically those that are trained in an instrument versus voice, thinking of plasticity, if we can see like greater or lesser increases.
Yeah, that is a really great, there's another PhD thesis topic. If someone would like to do that, please come to my lab and we'll do that. I think that would be extremely cool, because what we're looking at and what I discussed is kind of global in the sense that we're not taking into account people's past experience. And the arguments are about, speech and music at a very global level, at the level of, what humans tend to share. But on top of that level, we know from lots of experiments, including some of like Shihab Shamma's experiments, where these spectrotemporal receptive fields are very malleable by experience, sometimes even over a short period of time. So he's trained ferrets to respond to a particular spectrotemporal stimulus, and he shows that or Jonathan Fritz, I think, did these experiments, shows that there's like a sharpening of the receptive field in response to what becomes behaviorally important for the animal. So it'd be very important and interesting, let's say, to take Floris's brain and play him, some oboe sounds, and look at the particular acoustical signature of oboe and see if he's got more, brain units tuned to the oboe, right? I think that would be a very cool experiment. I'm not aware that anyone's done it. So, PhD thesis?
Great. We have online also Jamie with a question.
Hi, thank you so much for your talk. My question's kind of it's similar to others, but and it's more about maybe something you noticed during your research rather than a specific thing about the research. So I said that if you're looking at different aptitudes related to your right or left side, either the spectral resolution on the right or the left side temporal resolution. So like if you have more precise differentiation between spectral information or more precise differentiation between more minute pieces of temporal information, you might say that you have certain musical aptitudes. Was there a correlation between the right side resolution aptitude and the left side that you noticed? And was it positive or negatively correlated, or was there seemingly none at all?
Yeah, that's a great question. Thank you.
Thank you.
The particular experiment that I'm focused on at the end, which is Philippe's science paper from 2020, we did not look at individual differences in that way. We did look, as I mentioned, at the pattern of errors in the confusion matrix, which we found matched their brain responses. But the experiment was not designed to elicit large individual differences, in the sense that when you took the undegraded stimuli, performance was really pretty good. It was a pretty easy task. And the sample, was not selected to have a large variability. On the contrary, we wanted a kind of a homogeneous sample. I think that the question is a good one, because I think the model, it goes back to another question earlier, the model does predict that the sharper your tuning is in one domain or the other, the better you should be behaviorally in one domain or the other, right?
Right.
There are behavioral differences between people. Some people have better acuity in some domain than another. So from everything that we've seen, I would predict that those should map on at some level to these neural responses. We do have evidence of that in some respects. Like, for instance, early in the talk, I mentioned the frequency following response. And if that's not looking at the spectrotemporal domain, but it's just looking at the frequency domain. But we do see that the greater the fidelity of the FFR to a tone in a person's brain, the higher their spectral resolution, or the lower, I should say, the smaller difference that they can detect.
Interesting.
So, and there are numerous other examples of that kind of accounting for variance in the population. So, yes, I think that is interesting, and it ties into also the plasticity question, because it may well be that if you spend all your time listening to the oboe, there may be that may come at some cost, right? And in my brain, it'll be the organ, and who knows what else I'm missing out on. Yeah, super interesting questions that I think are worth exploring. But I think the answer I'm pretty sure is yes. But what's nice now is we have this kind of framework. So
yeah,
we can exploit that, right? We can now
definitely
more precise questions maybe than we could before.
Thank you so much.
Thank you.
Thank you, everyone. So we're going a bit over time, so I would like to close up the official part here, also because we actually have some refreshments coming in shortly as well.
But I see Mark. Mark is here, and I would like to hear what he has to say.
Yeah, sure.
Hi, Robert. No, that's now I feel very honored. I was just kind of saying, oh, damn it,
Honored that
just
you
when I was ready to get the microphone. So very good to see you again. I'm trying to switch on my camera, but it's pitch black here now. Not really see something.
Hi.
I was wondering so you're framing this hemispheric specialization in terms of keeping our speech and music streams, disentangled. But what about the lateralization for communication sound processing in animals that don't have music?
Well, by and large, they don't have a lot of lateralization either, right?
No, it's not true. There are a couple of primates have it, and mice have a very clear lateralization for communication sounds.
Well, that's an interesting question then. It may well be, I guess my answer would be that to the extent that such differences exist, it's probably because there is some environmental demand to have good temporal responsivity and to have good spectral resolution. So to navigate the environment, I think it would be interesting to look at the ecological acoustics of the particular species, not just its communication, but maybe other sounds in its environment, maybe sounds of predators or other sounds, natural sounds that might be very relevant for that species. We know from Tunison's work and other people that the spectrotemporal receptive fields tend to be tuned to the features of the particular animal, right? So
yeah, thanks. I think it's a very good point to look at not only the vocalization sounds, but also the other sounds that are relevant for these animals. Thanks. Have a great evening there, everyone. Bye.
Thank you, Mark.
Thank you, Mark. Good. So with that, I would like to close this part. So please join me in thanking once more Robert for this wonderful talk and for being with him. And he's going to stick around, I think, for the reception as well. So this is your chance if you want to do one of these many PhDs that are on offer to secure your spot here. First come, first serve, I think. So thank you, everyone. And thank you, Robert, in particular. Have a great day.